All right, so I'm just going to do a quick 1-minute intro. This is our community analysis, microbial community analysis workshop today. And I'm Chris Henry and from Argonne. And we're going to be presenting 4 sessions today, starting with an intro of model seed, which actually would have been more appropriate in the other session, but we couldn't squeeze it in with everything else that was in the other workshop. So we're putting it in here. It's perfectly relevant here as well. So you'll get a little more background about the Model Seed plus Omega apps that you heard about last time, but with more of a focus on the model reconstruction side of things. Then I'll be presenting community modeling simulation tools, both the tools that we have today and the ones that we will soon have, the ones we were hoping to have today. And then Ranjan will be stepping up and presenting about pairwise analysis tools between strains. And then finally, Ulas will be presenting, is it Ulas or Gianna? Okay, Ulas and Gianna will be presenting on the microTrait and the tools from that. So, all right, so we'll start with Jose.
So, thank you, Chris, for the introduction. My name is Jose Faria. I'm a computational biologist at Argonne, and even though you already heard about these apps the other day, I think the reason we can still, it's important to go over, for me in the next, I think, 15 minutes, to go over this work, it's because this is the basis for the community modeling. There's no community modeling without the let's say, simple single organism base app, base model seed modeling. So all the community model builds on a model seed. So we're going to go over this with you guys today. So just a little bit of a pitch here. So this narrative, you should be able to find it, this JSP 2024. If you search, you know, you can see kind of my narrative navigator here. If you search for this narrative, it should show up since it's public. And I'm not going to ask anyone to be following along. I'm going to be moving too fast for that, but you can always refer to it. And this narrative, a version of this narrative is also listed in this publication, which I also recommend you guys checking. So I was also plugging in this preprint because this is where you'll be able to see all of the details, you know, all that is behind the scenes on these methods. The methods in details, the algorithms, you know, the GitHub repos, everything that is happening, you'll be able to be here. So I highly recommend checking the preprint out. Hopefully, we're hoping to submit later this spring. So I'm gonna get started. So first, I'm just gonna give us a little bit more space. I'm gonna hide my data panel here. And the Model Seed 2 app is for people that have been using Model Seed for now over 15 years. And this is the first significant update that we have done in those 15 years. We have progressively done minor updates through the years, but this is the first time that from the ground up, we have redone the entire pipeline, touched all the templates, introduced new templates. So I'm just going to give you a quick overview of what that looks like. We start with RAST annotation. You guys seen work before already on how we're now enabling you to use multiple sources of annotation, but you always have to do RAST. Even you may use additional ones, but RAST continues to be key. We use a machine learning predictor to try to tell you to select your template for you. You also can manually choose it. The big difference now is that we start with a core. We build a core model and we try to make it right. We try to make sure that the core model is able to accurately represent the energy biosynthesis of your organism, which is what it determines the majority of his lifestyle, before we expand to the genome scale. So I'm going to go over here. So the first step, as we talked about, you annotate with RAST. So I'm not going to go over with that. People ask why RAST? RAST because we have based our mappings in our templates historically over RAST. We're now expanding to other annotations, but we still believe that RAST, I'm not going to comment on the quality of annotations of RAST versus others, other algorithms, but I can comment on the consistency of those annotations. RAST is very good at annotating consistently with the same vocabulary without any, small string variations and weird things showing up in the protein names for the same proteins across genomes. So in a world before machine learning in AI was very important to have a more consistent vocabulary. Nowadays, I would say we're working on tools to enable us to overcome that, where we can more easily map between annotation data sources, because AI is able to understand, the semantic context of all of these different proteins and that protein functions, and that's something that it's coming down the line that is going to help further our annotation mappings to other sources. So pretty basic. You're able to build. You guys already seen this app, the build prokaryotic models and metabolic models with Omega. In this case, all that we did was build a draft model. We start here. And what is a draft model? A draft model is a model where you do no gap filling. Why is it helpful? Sometimes you really just want to know what's in your model before you introduce anything with gap filling. You want to know what's your basis. And you can then use that base model to do gap filling later on. So it's always good to have your draft model as a basis. To be honest, too, a lot of people, that's all that they want is the draft model. They're not interested in running simulations. So for us, it's very important that we give you a good draft model without gap filling. A lot of people just want the mapping between reactions and annotations. They just want to know the metabolic capabilities of their organism, and building a draft model is a pretty quick way of doing it. There's arguably they could use other tools for that, but it gives you a very quick way to understanding this. So part of the new things that we do is now you see these reconstruction reports, and I'm going to open this. So if you open the reconstruction report, this is where we're trying to give you a little bit more information now. As you can see here, he's telling you that no core gap filling was necessary, meaning when we build the model at the core, we didn't do any gap filling. It was not necessary, which is good, because it means that we're able to accurately capture the energy biosynthesis of your organism without doing any gap filling and possibly screwing that up if we don't do the gap filling right. And you get this kind of nice analysis here. If you look down, even though you say no gap filling was available because we didn't run gap filling, this is a draft model, but we do test ATP production against 54 different medias at this point. So basically you can see here that for glucose aerobic, there's 26 ATP production. Again, this is just at the core. There will be, once you go to genome scale, there will be other additions that will lead to the increased production of ATP. But just at the core model level, you can see that in this case, as long as one of these media is able to produce ATP at the core, we don't do any gap filling at the core. We don't have. If one is okay, it's like okay, let's just leave it at that. But as you can see here, but we also tell you, well, for example, here for this succinate O2 or succinate in general, these two, it says if you were to want this to produce energy under these conditions, this one reaction here is missing. So we're telling you that, and in this case, what is the point of this? You would say, well, this reaction, since it's just one reaction that needs to be gap filled for this to be able to produce energy with succinate, it makes you question, is it real? Then that's when you start making questions and maybe start using these Omega tools. Is this a real gap? Is this organism can't do it? Or is a gap in annotation? Or there's an issue with a modeling template that is not capturing this? But when you look at something like methanol, and you need 7 different reactions in order to produce energy, we're pretty confident that E. coli is not able to grow in methanol, I would say at this point, because you need to fill so many gaps that it gives you that information. You get kind of this negative result saying, oh yeah, no way this is happening. So and you can cite and you see this for other sources. So this is one of the big themes of these new versions is we try to give you more clues of what happened when you built the model instead of you trying to dig through tables and trying to understand yourself. So you're able to build your draft model. And then you can build your draft model in something like glucose minimal media. And I'm just gonna go over, again, this, I can show you the parameters of this app. Something simple, similar to what you saw already. You select your genome, you select your model. You're able to, you can see here for omega, you will be able to put your expression data sets, and you have, you select that you want to do gap filling. For the draft model, we uncheck this box here that is checked by default. You also see it here. So that's before we had it unchecked, now we have it checked. You can select the template yourself, but our machine learning does it automatically. And the gap filling types where we have a sequential, independent, and global, and this was also discussed on the previous workshop. I believe the global is currently not working yet, but it will be soon. But more important that I want to go over here again is this report. We think that report, it's really where people are going to find some novelty is when I look at this report. So here I'm telling you what is happening here. You get this gap filling analysis. Now that we did gap filling, that tells you what reactions were gap filled, but more important, tries to give you a reason why. So in this case, we see here that we have this exchange reaction being added for this methylthiouribose, and we have these reactions here, and we have also a symport being added for it because this is necessary for the biosynthesis of spermidine. In this case, E. coli needs its spermidine as one of the biomass requirements. So in order for that to happen, these reactions need to be gap filled. In this case, I know exactly what is happening. There's no reaction in a model currently that is able to recycle this compound in a model. So it needs to be pushed out. It needs to be synced out of the model for the simulation to work. So that's what it's doing here, is adding a transporter and an exchange reaction out of the model to make it work for this compound. Here, we see that it's a lipid reaction. So there's these lipids that are necessary that are, in order to produce those lipids, this reaction needs to be added, same thing. I believe in this one because lipids are hard to annotate. Sometimes there's some bad lipids annotations. And this is just in this reaction for as far as your simulation, it impacts nothing. It just completes the fatty acid biosynthesis. It has little to any impact. So this is us trying to give you clues about what might have happened. Imagine you say, look, I'm sorry, Jose, but like my organism doesn't require spermidine. So why are you gap filling for spermidine? And I was like, well, great question. We have a universal biomass reaction. But since you are the expert in your organism, you can go to the model, you added the biomass reaction, you remove spermidine, and then next time when you gap fill, this doesn't happen. So this also helps you trying to figure out, give you clues to manually tune more specifically your biomass reaction. So, and I think I have like 4 or 5 more minutes. So I hear this narrative as a tutorial. You can see here it has a lot of text. So basically all that I'm saying is probably better explained over text. When I wrote this, I probably put more thought into it. But the same thing. But again, you get the same, in the report, you get the same ATP analysis. In this case, you'll see that is exactly the same as it was before, because even though the previous in the draft, we didn't gap fill it, but it doesn't matter, right? Because this happens at the core before this genome scale gap filling. This step of checking for ATP production, it always happens before. You do see this one thing here, just a detail that where this, for this formate, there's this filter reaction. All that it's saying is that our algorithm is saying that if this reaction is present, it's going to lead to a infinite production of ATP under these conditions. So we filter it out. So this may this may be an issue with the reaction itself, or just a combination of reactions that it's leading to some weird, flux mode that can cause energy generation loops. So other people have used algorithms after the models are built to eliminate these energy generating loops. We try to do it at the reconstruction step. So, and then if you run flux balance analysis, you're able to get an FBA, and they went over that. And if you have a model and an FBA, you can use this Azure Pathway Viewer app. I just pulled this map over here, for example, for fatty acid biosynthesis. And you can kind of see the flux going through this map. And we have like a full genome scale map coming soon. But all that I did in this app, on the Azure app, which is in beta. So I had to, just a detail, I had to do this. If you click here from between release and beta mode, you'll see this Azure Pathway Viewer app. We'll be releasing it once some things are polished. I selected this BIOS, there's a bunch of maps that you can select, and you select your FBA solution, and then you're able to paint and get your flux solution painted in a map. So it's quite nice, and you can even export this map out. If you have expression data, you can also visualize it in the map as well, overlaid with your flux. So we think this is going to significantly improve visualization, because currently we would only give you KEGG maps. So then other things that you can do, again, this is just kind of explain you the basics. You can just, if you know what media for your organism, by default now, we let you build in auxotrophic media. So auxotrophic media is a media that has all proteins and amino acids, and the goal is to try to help you determine auxotrophy, these auxotrophies. And when you look at it, you can see that now there's less gap filling when you're feeding more things to the more rich media to the organism. But so you can see those differences. And since I need to wrap up in in 2 minutes, you're able to see that under a different media condition, now you have less gap filling. Another thing that you can do, if you guys remember from the past where we used to do complete media, you can still change that now. Complete media instead of auxotrophic media, where there's this flag here where you can change the default from auxotrophic media, which it tells you here, that's the default, and it tells you what it is, to complete media. Complete media is even the more rich, is the theoretical richer media possible, because it's the abstract representation of everything that the model can uptake. It's helpful if you're trying to assess the total possible capabilities, metabolic capabilities of your organism, so you can still do that. You can still, edit different medias, run multiple medias, and then the cool thing now is that if you do multiple medias like I do here, this report, the report will tell you, this is what I wanted to show you, for the different medias what happened, right? It's telling you here, hey, this reaction is being gap filled because it's both needed in this carbon glucose minimal media and this anaerobic alpha ketoglutarate media. So it tells you, and in some cases, well, these are only important for this media. So you get a list per media if you do multiple media of why, what reactions were necessary for each media. So you get that detail as well. And then the last thing I want to go over would be this highly requested feature, which is blacklisting reactions from the pool. So sometimes you look at one of those reactions, and you were like, "Why? I don't know. I don't like that reaction. Like, what happened? What happened in your organism?" So you're able to select reactions. It's like, I'd like to see an alternate solution. So because sometimes with flux balance analysis, all that there may be an equal path that could be chosen, and by random chance, the algorithm chose the one that you don't like. So you can now have the option to go here and select from our database. You can select, you say, well, this gap-filled reaction, ban it, run it again, and then you can get, if there's another solution in the database, you'll get a different solution. And then you can judge for yourself and your knowledge which one is the better solution for your problem. So that's another way, that's one of the most requested features we had for the last handful of years. So you can ban reactions from the results. So, and then the last thing I'm wrapping up now is you see that this parameter here, that is the about turning off this ATP safe gap filling that we described, basically the thing that prevents you from model to kind of going wild. This apply ATP check, you can turn it off. We don't recommend you do it, but sometimes there's a small chance where your models you're not going to be able to gap fill a model unless you let the gap filling run wild, and that can still be helpful. Like if you're not finding a gap filling solution, I recommend you can uncheck that box, and then you have to analyze the solution and really try to understand what's going on. Why is it that I needed this really, to uncheck this safety check in order for the model to gap filled? At that point, I highly recommend you really digging through the gap filling reaction and trying to understand what happened. But you can turn it off. I would not recommend it unless you kind of really understand, I would say, have a more advanced knowledge of these tools and have really understanding how to interpret gap filling results. And there's a small list of known issues at the bottom that we're working on. And some might have already been fixed since the new deployment of the latest Omega app. And that's all that I had for you guys today. Thank you for your time.
Okay, with that, I'm going to talk a little bit about the community modeling tools that we have available in KBase right now. So I'll be talking about a workflow we've done with the Grow team that we're hoping to be able to disseminate more broadly throughout the community. So if this is interesting to you, not all the tools that I'm going to talk about right now are deployed immediately on KBase, but many of them will soon be deployed. But if they're interested in what I'm presenting, let us know and we can work on applying these tools to your systems as well. Even now, so we'll just have to help you offline a bit. We're implementing this diagram. We call it the mechanistic ecology diagram. So we've gone over this with the SFAs and the BR community in the past, and it really captures all the steps from raw reads to ultimately reaction transport models, which we're not there yet, but we want to. A lot of us have that goal of getting there. And Jose talked about this first step of being able to turn MAGs into energy accurate predictive models. And now I'm going to talk about this other step of actually doing community simulations, so combining models together to do a community simulation. So we applied the Model C2 to build models of the Grow MAGs, and there were, So we applied the Model C2 to build models of the Grow MAGs. So we get many different, we can build this model dashboard. Again, this is something I hope to put in KBase very soon, because really the Model C2 app, it runs in bulk, it runs on multiple genomes at once, it gap fills multiple medias at once. It's designed to process massive amounts of data. And so we want people to be able to build models of thousands of genomes or MAGs, and then look at the stats from those models. But the issue you see is in this figure, we've got the check-in stats for the MAGs, and you can see the completeness is all over the place. It's cut off at 50% because that's their quality metric they use. So they basically didn't release MAGs that were lower than that. The contamination is generally pretty low, but we see the completeness is all over the place. The genes are all over the place, and this is a mixture of diversity and the quality of the MAGs. And then we can see the modeling stats, and in particular, the gap-filled reactions, which are in green. And you'll notice a little bump in the gap-filled reactions here. And this is because this clade here is really weird and not being annotated well. And this is why this dashboard is useful. I've seen people build models, and when you build models at this scale, it's easy to miss that something has gotten messed up. And so it's really important until we give you this app, please get the stats and make this graph yourself. But I'm hoping to give people this app soon so that they can rapidly build these dashboards. But this is the stats when we applied the Model C2 to 5,000 reference genomes, and this is the gap filling. So compare the green line here to the blue line here. There's a lot less gap filling on the reference genomes. So MAGs have this issue. We'd also applied the Model C2, we get the energy pathways. You can see there's more gap filling, so that the green means there's zero gap filling for the core metabolism. So again, with MAGs, we have this issue, we have more gap filling, and we need to deal with this. Like the models, I'd say MAGs today generally aren't good enough to do quality community modeling. So how do we deal with this issue? And this is the way we've come up with to deal with this issue. We build what's called a probabilistic model. We talked about probabilities a lot in the last session, and those were probabilities that an annotation is right. And those are very important probabilities and should be integrated into this framework. But here I'm talking about something different. Here, remember, we're talking about dereplicated mags. So GROW has produced 2,000-ish dereplicated mags across all their samples. I want to be clear, these mags are from one sample. They're dereplicated because they had a high ANI score against mags from another sample. So we're already, I just want to be clear, like we're already making an assertion that this mag is representative of this strain across multiple samples, right? So we're already making an approximation here. So all we're doing here is we're saying, okay, this mag, you can imagine it almost like an ASV, but a really good ASV with a high resolution, much higher resolution as you get just from 16S. This is a whole genome or 50% of one. And so I can dial into a very accurate space in the genome space, the evolutionary space. If we were to lay out all genomes, all of our isolates, all of our mags in an evolutionary space, and then search for similar mags and genomes. So all of the GROW mags, we eventually want to have, and when we get our CDM built, all mags that any user can see. So you won't see private mags, but you will see any mag anybody's made public. You'll see it, and it will be integrated in this analysis, and the person who made those mags will get credit. They'll be linked to this because they'll get credit for the fact that you used their work to make your enhanced mag that now encompasses all the genomes in a circle. And the key thing is we can make this circle broad because a lot of times, and I'll show you in the next plot, you don't get many hits. You know, for some of these really weird mags, you just don't get many hits. You don't get any hits among isolates, and you only get hits among mags from environmental samples that are close to you. And that's why we think it's so important that we pool all the mags everybody is seeing live, because, you know, it's the live, immediately shared work. The frontier is where people are commonly like looking at the most important and least understood systems at any moment in time. So we aggregate this, and what we do is we compute a probability that any given function appears in any of these mags. If it's in all the mags, it's a core function. It has a probability of 1. If it's in the mag that you're actually querying the database with, we give it a probability of 1. You have definitive evidence it's there. If it's in only one of the, you know, in this case, 3 mags, it has a probability of 1/3. So that means, okay, it might be in your mag, but, you know, it appears to be part of the flux, and it has less of a chance of being in your mag. It has a 1/3 chance of being in your mag. And so what we do now is we simulate these probabilistic models. We'll still give you a mag model that has the core, that only has the 100%. And that can be your mag model. It'll be better than the models you're getting now because it'll be enhanced by the core of the pangenome. But we are also going to make what we call a probabilistic model that will have the core and the flex with probabilities. And then when we run FBA on the community model or on that model on its own, we use these probabilities in the FBA. We say, try not to use that flex. Replicate this behavior that I know my mag has, but try not to use that flex genome. Try to just use the core and the highest probability stuff, because that's the most likely stuff that's actually in your strain. And then we can also, if you have expression data, we can invert those probabilities and say, if you have expression data, if you're actually seeing this gene expressed, yeah, try to use that reaction. So try not to use the low probability stuff. Try to use the stuff that's expressed. The more it's expressed, the more I want you to try to use it. So that's the simulation approach we're building. We're not quite there yet. I'll show you what the app will look like. So I'll actually show you the prototype of the app in KBase in a moment, but that's what we're going for. What's nice about this is it integrates everything we know about this mag in the entire universe. It has the whole database embedded inside of it, and integrates the energy strategies of every mag in your community. It integrates ultimately the annotation probabilities will be integrated into this as well. So we can actually consider alternative annotations here as well. It integrates the metabolic capabilities of every single organism, the energy capabilities, the abundances, and the expression data. The other thing that's important about these probabilistic mags, you might be doing proteomics. If you're doing proteomics against a mag, you're going to miss stuff because they map their peptides based on the proteins they see in your mag. If there's a protein that's missing from your mag because your mag is 75% complete, they are not going to map the peptides they're supposed to map. We would argue that we could actually put the sequences of the genes that are close by into this, and the probability would give you a measure of how close that sequence will be to the sequences in your mag. So we can put the sequences in here as well, and they can use those sequences to map peptides. They could use those sequences to map metatranscriptomes with a greater deal of accuracy than if you're just trying to use the mag by itself. So this is an extension. We are making this phylogenetic functional approximation, but we're doing it with an acknowledgement of the uncertainty that is inherent in doing that. This is just a graph. It's kind of hard to parse now, and I'm probably over time, but this is a graph that shows how many hits we get in GTDB with the different sets of mags. And essentially, so if you start with 100% Annie, this is how many mags we have. As we decrease the average nucleotide identity that we allow to use in our clustering, we get more and more consolidation of mags and more recruitment from the GTDB database that we're mining against for the isolates. Again, this is a pretty laborious process right now, but once we build the CDM, this will be an essentially instantaneous process. Because as soon as you load data into KBase, it will all be mapped. It will basically just take your mag, look for the close mags and isolate genomes, check which ones you can actually see on the system, and then give you your output. So everybody gets immediate access to all data that's shared with them or is made public on KBase. So our workflow for Grow, again, we annotate the mags, we consolidate them into these clades. So that's so for Grow, for this Grow analysis I'm talking about, we can also do this at the clade level and just say, I know I have mags, I know they're poor resolution, I'm going to just group them at a particular phylogenetic level, in this case, the species. And so they took all of their mags that were a particular species and merged them into the clade. This was beautiful, and I recommend this for other people who are looking at similar like complex soil systems, because it cleans up the abundances and it cleans up the expression. Because at the moment, with incredibly complex soil systems, that data is extremely stochastic. Like it's kind of a matter of luck with what you see in the meta-T, for example, because you're doing metatranscriptomes against this incredibly complex soup. So they build these clade models. We apply the model seed with all of its, wonderful energy analysis and gap filling. And then we merge these into a community model, which we then simulate with our mag abundances, our gene expression, and our probabilities. So same probabilities apply with the clade model, except there's no master mag anymore. Everybody's got a probability, a lot of the reactions have a probability less than 1. And then we get a clustering of the output, and I'll show you what that looks like in a minute. The pathway rules, the pathway fluxes through each individual clade or species in that mag, the actual interactions between the clades and the environmental interactions. So that's all the things the model tells you. And if you build a clade model, it does it at the clade level. If you build it with mags, it'll do it at the mag level. This is what we've done here is we've taken the pathway fluxes through all the clades, and we've run the community model, but then we've broken out the pathway fluxes into the individual clades, and we've clustered them. And each color is a different clade. So what you see immediately is they cluster almost perfectly. We didn't try to cluster to make the clades cluster together. They just did. So what this is saying is with the model seed modeling approach, with the clade approach, with the merging of the genomes, we're getting good enough genomes that everybody has a niche, and that's that space. And sometimes, because of the abundance and the metatranscriptome data, you have species that hop out of their niche and they do something different for a couple of samples. They kind of step into somebody else's niche. So you actually capture everybody's individual niche. Everybody's doing something different. They're all interacting with one another. And we can dig into what's happening in these, like when the blue clade gets split, what's going on in these samples versus what's going on in these samples. All the data is there for us to dig into that ecology that's happening in these systems and dive into more detail. All right, so I want to acknowledge the team and particularly the GROW, and this was funded by both the GSP and the CSP, so the Community Sequencing Project from JGI, of course, but also the GSP-funded project from BER. And I'm going to over time a little bit. So I'm going to just quickly show you the narrative. So there's an introduction. This is a public narrative, and we'll link it in the documents, or I can stick it in the, or maybe Alicia can stick it in the chat. First, you merge the models. This app is, I recommend using the beta version of this app. We've seen issues with the prod version, but the beta version is working. You can merge the models. So again, I have 6 clade models here. So 6 different clades that we chose. There are more than 6 clades. We could put more clades in. We were zooming in on these 6 because there was interesting variation. There was interest in ecology. So this is another strategy. We made 6 clade models, and then we made another model. So I took all of their other mags and merged them together into an other model. The probabilities are low on that model. So it's not going to use a lot of stuff in other if it can avoid it, but it will use it if it has to. So this lets you capture it. This also gives you a mechanism for zooming in on a specific piece of the ecology that you want to learn about. You've got the whole system covered, but you can zoom in on a particular set of organisms and say, I really want to dive into how these guys are interacting. I see some correlation. I want to dive into that. So this lets you do that. So all those clade models are here with their probabilities embedded in the models. It's part of the reconstruction process. And then you can run the new community FBA app will let you put in expression data. The expression data, there's no community expression KBase. The expression data would be associated with the 6 clades, so you would just load the 6 clade expression data here. We'll probably make an expression data set so we can make this a little easier. You don't have to select them one at a time. You specify reactions you want to maximize. Generally, it's the community biomass we maximize. You can turn on and off this probabilistic FBA, this minimization of fluxes based on the probabilities in the clades, although I recommend you leave it on. What it does is it maximizes the biomass of the community, and then it sets it to some reduced value from the max, like 90%. And that's just to give the community some wiggle room to do interesting things as driven by your metatranscriptome data. So meta-T says, yeah, but you should be eating this, and you should be excreting that. So like it tells the model to do some different things. You can put in specific abundances and constrain the total resource for your community. So it's very important at community modeling, particularly if you're giving it a rich media to restrict the carbon uptake. But this app isn't quite, I intended to demonstrate this app today, so we're very close to getting this app out, but it's not quite running yet. But you can run the classical FBA app. It's just at the moment it gives you the fluxes for everything in your community, and it's mixed together. It's compartmentalized, but it's mixed together. So you have to like get these fluxes downloaded as a TSV and then compute the exchanges and things yourself. But in the new app we're building, it will do that for you and actually give you your interaction fluxes and everything in the output directly, hopefully with an interaction diagram as well, like a picture of how your species are interacting. So that is coming. And for that, I'll hand things over to Ranjan. Yep.
So hello, everyone. My name is Priya Ranjan. I'm a scientist at Oak Ridge National Lab. And I'll be talking about some of the community modeling efforts we are taking in PMI. So as part of this collaboration between PMI and KBase, we are building a set of apps that can help with synthetic community design. Now, the Plant Microbe Interfaces project at ORNL is interested in understanding the diversity and functions of mutually beneficial plant microbe interactions. And as part of this effort, I don't know if you can see this, yeah. The project has isolated more than 3,000 bacterial isolates, and more than 500 of these isolates have the sequencing was done by GGI, and so the genome sequences are available. And also, as part of this project, there are several experiments done on several of these isolates, which includes biolog data, inhibition assays, pairwise interactions, co-cultures, and so that's available for several of those isolates. But if you just imagine 500, and if you're doing synthetic design, like how many can you test experimentally? Even if you have 10-member communities, it's a very huge space. And how do you prioritize? How do you go from that large number to a prioritized list, which you can then put in your testing mechanism? And so we are leveraging machine learning and metabolic modeling framework in KBase. And right now, predicting pairwise interactions in simpler media, as well as phenotypes, with the ultimate goal of designing synthetic community. So the apps that have been developed as part of this project we have 3 apps. The first one is Calculate Metabolic Interaction Scores, which gives you scores that are a measure of cooperation and competition between the microbes. And then 2 apps, we have one where we wrap anti-smash, and the second one which predicts antimicrobial activity, which sort of gives you the inhibition potential in the pairwise interaction. So the first app, CommScores, there are several scores that Andrew here has developed. And so, but I'll just go into a little bit detail on the 3 of the scores that are there. The first one is metabolic resource overlap, where basically it's the fraction of metabolites that are overlapping as nutrients between the microbes. So this gives you a measure of sort of competition. You can think of it as some sort of competition. Then metabolic interaction potential quantifies syntrophic exchanges. And basically it's like where the metabolic product of one organism becomes the nutrient for another. And it's a measure of cooperation. And then the growth yield difference is basically the difference in growth yield between 2 microbes. There are several other scores, some of which are published and Andrew has curated and implemented as part of this effort. But then there are also several new scores that have been developed. The second app, annotated BGCs, Alexis and Dilip have been part of this effort, as well as predict antimicrobial activity. So anti-smash app, basically we wrap the anti-smash tool within KBase, and it identifies biosynthetic gene clusters, which are related to natural product formation. And some of these products can include antimicrobial activity. And we have rich genome viewer now within KBase to explore the data. The BGC antimicrobial activity prediction basically uses training data and classifier from a recently published paper. And within KBase, if you upload a new genome, you can use this, the machine learning framework, to predict the antimicrobial activity of any new genome. So all the apps that I have shown are currently in beta. And so that's where you can access them. And I'll quickly go through the narrative. Obviously, you start with a set of genomes. And then we have used this built prokaryotic metabolic models with Omega that Chris just talked about, as well as we had Jose talk about it. So I won't go into that. I'll start with the first app, CommScores, and it basically takes a set of models, or even model set, object type, as well as the media in which you want to evaluate your interactions. And there are several other scores which are meant to constrain the kind of interactions, the interaction pairs that you want. And so the result for this is, it looks like this, where you have each row. Let me go here. So each row here is a pair of model, like Rhizobium and Pseudomonas, and then the media in which this is being evaluated. Then there are several scores for model growth, model 1 growth or model 2 growth, MRO for model 1, MIP scores, and several other scores. There is a metabolites tab in this report where you can look at what are the syntrophic exchanges that are happening, what are the costless secretions that are possible. Then there is also a heat map view for it, so it gives you easy to compare different scores across different pairwise, different pairs. And then there is a documentation on what these scores mean, what's the algorithm behind these scores. Some of these are being updated, so if you have questions, do contact us. So that was the first app. The second app, annotate genomes with biosynthetic gene clusters using anti-smash. So it takes, again, a genome set or genomes as an input. There are several parameters where you can add more information, like compare with known clusters in MIBig database or save genome with the features. And the first view you get is a tabular view where each row is a genome and your columns are higher level categories of the secondary metabolites that are predicted by anti-smash. And you can look at the count of different categories in each of these genomes. You can also it also gives you a rich HTML report of each different biosynthetic gene clusters, where they are located, what they're producing, and the standard anti-smash output that you get, you cannot do within KBase. The third one is the antimicrobial activity prediction app. It takes, again, a genome set or genome as an input. And the result that you get is for each genome and each BGC, the biosynthetic gene cluster, what is the probability of it being an antibacterial or antifungal or anti-gram positive, anti-gram negative. It gives you this probabilities. So you can evaluate which of your organisms have more of that potential. So these are the three apps that we have developed. Now, I was trying to interpret, like, no, not this. So PMI, as you told you, it has synthetic community data set. So we took one of the 10 members community. And this is relative abundances of the 10 bacterial strains after sequential passages in R2A media. And so like this is Pentuia, this is Pseudomonas. And so we're looking at what do we see in the scores for these two most abundant, yeah. Okay. So CommScores predicts highest metabolic interaction potential and second highest community growth for this pair. We are waiting for experimental data where we can now take these scores and use it in a machine learning framework to predict 120,000 interactions. We have that data coming. So with that, again, the future work would involve building that machine learning framework, experimental validation, and make the apps available and release more. And with that, PMI team, thanks to PMI team, Iowa State University, KBase, Andrew for developing this course. If you have questions and you want detailed discussion, Andrew's here. and DOE for funding this. Thank you.
Great. So we will switch scales a little bit and talk about trait-based microbial community modeling in KBase. My name is Gianna Marschmann, and I'm a postdoc at Lawrence Berkeley National Lab, and I'm presenting today with Ulas, Karaoz, also at Berkeley Lab. So the trait-based modeling apps that we're developing are part of the Microbes Persist SFA, led by Jennifer Petrich. So the way that we currently use genome-informed trait-based modeling is essentially as a way to funnel some genomic information into larger scale models. So, for example, EcoSys is an ecosystem scale modeling, or RESOM is a dynamic energy budget model that's currently being integrated into the E3SM land model. The way we try to do this is by reducing the dimensionality of genomic data by inferring traits from genomes and then clustering these into microbial guilds. That we then simulate how these guilds compete across sample-specific environmental conditions and use the output of these simulations that could be guild-specific growth rates, calm use efficiencies, substrate uptake kinetics, or respiration rates, even to basically inform larger scale models. So we'll generate parameter distributions that have a representation at these larger scales to run a sensitivity analysis. However, these tools are still in development, so we would really like to get your feedback on where this could go in KBase. Formally, these trait-based models are part of a continuum of models, but in reality, we currently don't really have a connection to the representation of a metabolic network. So to do that, we would need some way to abstract a metabolic network. We would need to know how to cluster chemicals or substrates at ecosystem scale. So in the way that we reduce the dimensionality of genomes into microbial guilds, we would also need a way to cluster chemicals into chemical guilds at these scales. But today we'll go over two apps that we're developing for genome-informed trait-based modeling. That is, on the one hand, microTrait, which is a tool set that uses HMM models to infer traits from genomes. And then it defines microbial guilds and guild-specific parameter sets. And then we use these guilds, and then we simulate these guilds and how they compete in a coarse-grained representation of carbon and energy allocation in a dynamic energy budget model. So these tools are published, and we are working on a public narrative. So Ulas will go over the narrative next.
Hi everyone, my name is Ulas, and so this narrative that we've been building is on app tab right now because our apps are not in beta yet, but we'll make this available when it's ready, the same narrative on the main server. So as Gianna said, we are building two apps to make trait-based modeling possible in KBase. So before describing the apps, I want to talk a little bit about the data set that we are using here, which is from Kate Zalnina's work, published in this publication. So Kate's question was to understand the mechanisms by which the microbes respond to signals from the rhizosphere. So to do that, she isolated a pretty large set of isolates, about 300 isolates associated with the bulk soil from the Avena, and then she measured the abundances of those isolates as the Avena root grows. And she then picked a subset of those isolates depending on their phylogeny, so to broaden the diversity of the isolates, and also depending on how they responded to the root growth. So those 39 isolates are shown here. So this is a bar chart where you have the 0 point here on the right side. So it's sorted by whether each isolate on each row, how it responds to the root growth. So all the things that are on the right, they're the isolates that responded positively to the root growth. And then on the left are the what she calls the negative responders. So we are using this data set in this narrative from this 39 isolate. So now I'm gonna sorry. So now I'm going to switch describing the two apps that we are building. So the first is called microTrait, and the second is the dynamic energy budget modeling app that we call DEBmicroTrait. So just to tell you a little bit about the microTrait. So the microTrait is an HMM-based pipeline that focuses on the traits that goes into the trait-based representation of the microbial genome. So we focus on ecological strategies of microbes that can be directly used to parameterize trait-based models. So those traits fall into three categories. The first category is the resource acquisition. The second is the resource use, and the third is the stress tolerance. And there's a hierarchy below those categories that captures those traits. And I invite you to read the paper if you want to know about the details. So essentially what we are trying to do is to enable trait-based modeling, we need to reduce the complexity of the genomic data in a quantifiable way. And that's what we are doing here. So the app takes as input genomes, or if you have assemblies or bins that you think represent genomes, they're welcome too. And then the only parameter right now that we make public, it's the amount of variance that you want to keep under control to define the yield. So that's the inter-yield variance to define yield. So as you increase this number, you're going to end up with more granularity in the yield. And so we run in this narrative, we run this app for this 39 genome that I described from Kate Zanlina's work. And you have two major outputs. So the first one is the trait matrix, which should be obvious to you what it is. So it's essentially a heat map of genomes and their trait profile. Again, like these are all the traits that fall into those categories that I'm not going to go into detail here, but you can read it in the paper. And then the important thing here is that we also measure the trait variance across genomes, so in a quantifiable way. So you can pick a point here and then define the yield matrix that falls from that point in the distribution. Now I'm going to switch back to Gianna to talk a few minutes about the dynamic energy budget model. We are a little bit over time, so let's give her 3 minutes. Okay.
Okay.
You're good. You can use 5 minutes. It would be great if you can use 5 minutes.
Yeah. Okay. So for the second part of the app, we identified common root exited metabolites of a model plant species, and we import these specific compounds into the narrative. So we use the import chemical abundance matrix app, which just imports a CSV from the staging area. So what I was alluding to earlier is that here you see an example matrix. We basically use differences in stoichiometry, so the formula and mass, so molecular weight to determine diffusion constraints, for example. But we would need we currently inherit the chemical class from actually the transporter description in microTrait. So ideally, at this point we are tied to a chemical abundance matrix or a media type, but we would like to find a better way to classify chemicals in KBase. The model at the current point uses a slice of the trait matrix, so it simulates a few resource acquisition, resource use, stress tolerance, and life history traits, and it can be run in different types. So in batch mode, it simulates it gives a global picture, so it simulates how guilds, depending on the trait variants, grow on all substrates in the trait matrix. It can be run in mixed mode, where one guild grows in a mixed media or in competition with N guilds on M substrates. You can check on the KBase medium. So microTrait inherits the trait matrix from DEBmicroTrait inherits the trait matrix from microTrait. It gives a chemical abundance matrix, and then with null input for the interguild variance, it runs simulation across all genomes or guild genomes in the data set. The output currently looks like this. So we have distributions of substrate kinetic traits, Vmax and Km across these predefined substrate classes, global distributions of growth rate and carbon use efficiency, percent uptake from the mixed medium across these substrate classes that could be compared to, say, exometabolite uptake. And then you see that different guilds in this case prefer different broad types of substrate classes in the growth medium defined from Avena exodates. So future developments would include expanding the DEB app to accept more parameters, input parameters from microTrait than the current slice, and also to improve the format of the model output, so to actually output guild-specific differences with the app. And then we are thinking about having a better chemical classification tool for the representation of chemical guilds at ecosystem scale. Okay, so with that, I'd like to acknowledge the science part that was done at Lawrence Berkeley National Lab and Lawrence Livermore Lab, and KBase support for implementing the first version of that narrative. Thank you.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.